Hello everyone, this is your pre-lab video for the 8th week of the semester, the week of March 7th. Just a couple of announcements before I get started. Just a reminder that there are no shorts or open-toed shoes allowed in the lab at any time. The reason for this is because we're working with chemicals that we don't really understand the biological properties of, and we don't want them or anything sharp falling on your foot or on your legs while you're working. So no shorts or open-toed shoes. You will not be allowed to participate in lab if you wear either of these things this week or any week for the rest of the semester. Compound testing is going to begin this week, and like I said, the compounds that you're testing, we have reasonable information to suggest that they have some properties that might make them useful to treat cancer. And as such, they could interfere with critical cellular functions. So even if you're studying something that's an extract of something natural, you should treat these things as if they are toxic. So treat them in the same way as you would colchicine, wear gloves, and be very careful. So nobody should be working with any of the compounds without gloves on. This week in Discovery Lab, we'll be talking about research that's performed in Tintin Su's lab. And again, as a reminder, this is the research that we are performing. Tintin Su developed the strategy of performing a compound screen in Drosophila melanogaster to identify chemotherapies and has been fairly successful with this. She has started a company that brings compounds even closer to clinical trials to become new chemotherapies. In the lab this week, you're going to start compound testing, but this is going to involve a lot of different procedures because we'll be maintaining the population cages, you'll be isolating third instar larvae, and you'll also be setting up your vials where the larvae will develop, and those are the vials where you'll be quantifying survival in the next couple of weeks. This week, you don't have any formal submissions for your research project. You've already submitted your proposal. Those proposals were peer-reviewed last week. And this coming week, Gretchen and I will be compiling the scores for your proposal and the peer reviews that you received, and we'll distribute them to your group so you can take a look. You don't have to do anything with the peer reviews, but I think it's really important to take a look at them to see how someone else would have perceived your proposal, the strengths, the weaknesses, and it gives you an opportunity to take that information and improve on your project. The next thing that will be due as part of your research project is a draft of your introduction, and this will be in, I think, another week's time. But we'll be giving you rubrics. Those are already online if you want to take a peek at them, and we'll be describing to you how you'll be writing the introduction section of your poster. If you are feeling a little bit rough about the procedures that are being performed, you can look on Canvas. In the course schedule, there are video protocols that you can watch. We reviewed a couple of them last week. The first is the embryo collection protocol. You'll be doing this in every class for the next few weeks as we maintain our population cages, because those embryos become the source of the third instar larvae that we use for our experiments. So you'll be doing that in the first and second lab of this week. You'll also be collecting third instar larvae using the sieves, and you'll be setting up your food drug vials. And these are the food and drug vials that will contain the drugs that you've chosen for your research projects. Most of the compounds have come in. In the lab, when you arrive, you'll see that you and your group will have one box. Upstairs in A1B 18, the boxes are blue, but the boxes have all of your names on them as well as the name of your section and your compound. In each of the boxes, you'll find the stock solution that we prepared for you. You can ask your TAs for more information about this. It should say the solvent on the tube and the concentration, and your TA will help you understand how to get that compound into the food to test at hopefully a reliable concentration. For people who didn't receive a compound, I think there are 4 out of the 24 groups haven't received their compound yet. They're coming, so don't panic. What you're going to be doing is setting up more negative controls. We are going to need an enormous amount of negative controls in order to get a good sense of how the larvae develop when they're just exposed to a solvent. Almost all of the compounds at this point are dissolved in DMSO, but there are a couple that are in water. So we're going to be setting up more negative controls over the next few weeks in both DMSO and in water. So there are no assignments due on Sunday, March 14th, because we have an exam on March 17th. This week, keep an eye on the Exam2 study resources page. We're going to be populating that with some critical thinking exercises for you to get some practice, and also a practice exam. So you should be interacting with those, being active with your learning. And then the week of the exam, I think on Monday, the 15th, we'll have a review session, and I'll poll you all to see if we can come up with a time that will work with the most people possible. That review session won't be like the one last time where we go through the study guide, because the feedback that we received from you suggested that the study guide really isn't the problem. I can go over things that seem to be challenging on the study guide, but we'll be focusing our time on doing some critical thinking problems. So you should come to that review session having done the critical thinking problems so that we can review them in a productive way. So this is an exciting week. Have fun, and let us know if you need anything or if you have any questions about your projects.